Okay, cell biology fans, here we go. We're going to do extracellular matrix. And right off the bat, all right, we're talking about extracellular. This will be the first topic that we've really discussed that is completely outside of the cell. All right, these are molecules that are made by cells and they are exported through the process of exocytosis. And they are what cells sit on and interact with outside of the materials that are bound to cells, either as transmembrane proteins or as peripheral membrane proteins. These are neither of those. These are extracellular matrix components. Some of these are going to be proteins. Some of these are proteoglycans. Some of these are, they're called glycoproteins. And so you're going to see there's a lot of sugars and a lot of proteins that are involved in these extracellular matrix molecules. One of the things we're going to talk about are how cells contact one another, cell-cell contact, and also cell extracellular matrix contacts, because we already mentioned these two things, cell ECM contacts. And remember, these were two topics that were important for cell junctions, all right, but they're not just important for cell junctions, they're also important for how cells interact with the extracellular matrix. If I show you these two images again, remember I showed you these before, on the top left we have an image of a fibroblast. So this big cell in here that I'm pointing to with the red arrow is a fibroblast. And fibroblasts often sit in connective tissues, and those connective tissues have a lot of extracellular matrix molecules. In this EM image, you see a lot of white spaces, okay? That white space isn't space at all. There really is no space. And okay, this white space that is showing is probably made up of sugars. Okay, and sugars do not show up well on EM. And then the other molecules that you are seeing are what we call fibrous proteins of the extracellular matrix. So we have fibrous proteins and we have sugars. Okay, interesting. All right, these are collagen fibrils, and this is a EM image, a transmission EM, TEM image. So the cells are cut through, and what that means is you're seeing the longitudinal collagen fibrils, but you also see these little dots. Each one of those dots is the end of a collagen fibril. So these collagens are in bundles and they're perpendicular to one another. Some of them are coming directly out of the screen at you, perpendicular to those that you can see longitudinally across the screen. So this fibroblast is sitting in sugars and fibrous proteins. The image, the EM image on the bottom right, that's a scanning EM, an SEM. That image shows lots of cells here. So you see lots of cells sitting, okay, with this material that's on top of them. The basal lamina is here, and you have a lot of these extracellular proteins, these fibrous proteins, outside, and you can see those. They look sticky and they're cross-linking cells to cells and cells to the basal lamina. But a lot of this, right, since this is an EM image, you're not seeing the sugars. So a lot of this image is missing. But you do get the idea that cells sit somewhere. They're not just floating freely in the body. That doesn't work. They're actually in a matrix. Think of it as a sort of gel, and the cells sit there and are held in place by the fibrous proteins as well as the sugars that they interact with. Once again, okay, on the left now you have the scanning EM, the SEM. On the right you have the TEM. And on the right in the TEM you can see the spaces. On the left it's harder to see the spaces, but in both of these images what we're talking about is cells that are in a tissue are interacting with one another and with the materials that surround those cells. Right, so on the scanning EM, you can see some of these cells pretty clearly, but they're covered in extracellular matrix proteins and sugars. On the right, you can see the spaces, that's where the sugars are, and in this case, on a TEM, you may not really be able to see, depending on the magnification, the fibrous proteins of the extracellular matrix. We talk about two different things. We talk about the sugars, the polysaccharides, and the fibrous proteins. And we're going to talk about these with respect to how they function to hold cells in place and to hold molecules, proteins, or molecules that are secreted from cells in particular places outside of cells. So we'll start talking about the sugars, and as I said to you, there are proteoglycans, so that's proteins that have sugars on them. There are glycosamino, amino means that these are sugars, glycans, but these are really sugared up. They have sugars on both sides, glycosaminoglycans, and these are both part of the polysaccharides that are important for the extracellular matrix. The fibrous proteins that I've already mentioned to you, collagen is the big one that everybody knows. Some of the other, elastin has an obvious function, fibronectin, laminin. Okay, be careful here, folks, because today in class we just started talking about lamins. Lamins were intermediate filaments found right on the edge of the nucleus. That is completely different than laminins. All right? Good. So let's keep moving on. GAGS has a really great name, right? GAGS, like you're gagging, but what the GAGS do are they're the sugars, glycosaminoglycans, and in this image on the bottom of this page, what you see are sugars, right? So if you look at this boat structure, that's a sugar. This boat structure is a sugar. This boat structure is a sugar. So these are sugars, and some of them have amino groups attached, right? So can you see the amino group here? If I pull this out, it's got an N, C, C. It's not exactly the same as what we drew before because this is an NH with a C double bond to an O and a CH3, but this is called an amino group because of this amine at the end. So this is the amino part of the glycosaminoglycans, all right? And that's one important part of this because it creates this structure. But the really, really important part of this is the charged groups that are attached to these sugars. There are two charged groups, sulfates and carboxyl groups. So a sulfate group shown here, right, an SO3, which has a negative charge, and this carboxyl group, a COO minus, right? You should know how to draw those structures. Those are very, very important because of the negative charge they provide the GAGs. So think of the GAGs as the most negatively charged molecule in animals. Animals have lots of things that are negative. We talked about the DNA is negative because of the phosphodiester linkages between every single nucleotide. These glycosaminoglycans are the most anionic, the most negative molecules in animal cells. They're these repeating structures of sugars that have negatively charged groups on them, sulfates and carboxyl groups. Why do we care? Why does anybody care? Well, it turns out you really do care. The sugars and their negative groups, so we have these negative charges, lots and lots of negative charges, attract positive ions. And the most obvious positive ion that gets attracted is sodium. And so now you have lots of sodium being attracted into spaces between cells. And what is sodium? Sodium is an ion. And anytime you have a concentration, a high concentration of any ion, all right, especially one that's got a charge on it, we never like high concentrations of ions anywhere. Ions are not, right? We don't want to have a big pile of ions, and so water will follow anywhere there's a large concentration of ions to dilute out that large concentration. Concentration is, right, the number of sodiums per unit per volume. So sodium based on the volume. In a very small volume, if you have a lot of sodiums, that's high concentration. If you have the same number of sodiums and you have a large volume, that means that the concentration has gone down. So water follows wherever you have the sodiums. The sodiums are going to where the GAGs are, and that creates a pressure called turgor pressure. I don't know if you know about turgor pressure in plants, but plants, the reason plants stand up is because they have turgor pressure. In plants, the xylem and the phloem move nutrients and water back and forth through the plant. And so have you ever seen a plant when it needs water? It's kind of limp and hanging over. Well, you give it a little water, and then all of a sudden, right, that plant is, here's, my typical plant, is happy to stand straight up, and that's called turgor pressure. It's the pressure due to the swelling because there is water there. Turgor pressure occurs in your body in particular places such as your joints. All right, in your knee joint, in your elbow joint, in your ankle joint, in your finger joints, you have a lot of GAGs. And so those GAGs are in there. They're attracting, right, they're highly negatively charged. They're attracting sodium and water. And that then creates this turgor pressure that prevents the forces of nature from really causing you massive problems. Think of your knees, all right? If you didn't have all of this extracellular material sitting out there, you would end up with, okay, you know how I can't draw. Here's your femur bone, and it's sitting down on the bone of your what is it? Tibia, fibula. So in between here you have GAGs, and what happens is over time, as you get older, you lose some of those GAGs. You don't make as much GAGs, and you end up with bone rubbing on bone. And bone on bone happens because of compressive forces. What's causing that compression? Gravitation. All right, we're all stuck with gravity. Gravity is pulling us down all day, every day. And so that causes compressive forces. There's also forces of torsion, twisting, bending, and shear forces. We talked about shear forces with respect to keratin in skin cells. But really, compression is the forcing down on one another. Tension is when you have two things that are pulling apart from one another. Bending is pretty obvious, right? That's bending through the middle. Torsion is when you twist something. So the compressive forces of nature due to gravity are really, really tough on you, and the GAGs help protect you. You may have seen people that take this supplement, glucosamine and chondroitin sulfate. Well, guess what that is? That's GAGs. Glucosamine and chondroitin sulfate are GAGs. Here I'm listing for you some of the GAGs that are found in animal cells. Here's the chondroitin sulfate. There's also dermatin, heparin sulfate, keratin sulfate. All right, all of these sulfates have that really negative charge on them. Some of the GAGs will also have the carboxyl groups, which are also negatively charged. There is one important GAG that doesn't have, it's not sulfated, it's called Hyaluron, and we won't talk much about any of these, to be honest with you, but if people you know are taking this as a supplement, it's because they think that this really does help protect your joints. And so the real question about these GAGs, when they take them as a supplement, well, how do they get, or even saying joint ease, how do they get through your stomach and absorbed into your body in a useful form? And that's the big issue is, do they really work? Sometimes it's a placebo effect, but that's fine if that's really what the case is. But the GAGs, just remember, negative charge attracts sodium, sodium attracts water, water causes turgor pressure. That helps protect you from compressive forces. Now, the functions of GAGs and proteoglycans, right? So the function, right, is for protection mostly. After protection, what else do these things do? Well, if we just look down this list, immobilized secreted molecules close to where they are secreted. What's that talking about? Okay, that's talking about if you have a cell and there's another cell over here, and this one is secreting something. Let's say it's through the process of exocytosis, it's secreting a molecule. Okay, you're going to have a higher concentration here. It's going to be less as it gets further away. And that's because there's actually, right, this is an empty space here. Between these two cells, you actually have extracellular matrix, and they're trapping some of the secreted molecules as they're being secreted. Okay, this does multiple things. This can provide for a reservoir, provides there to be some proteins left later for additional use. It can protect the proteins from proteolytic degradation. It can actually bind to those proteins, the proteins that are secreted, and block their activity until something happens. And they're really good at creating concentration gradients. And I know we just saw the video of the neutrophil chasing down the bacteria, and that was due to the bacteria leaving, right, a trail of breadcrumbs, and that's a concentration gradient. The closer you get to the bacteria, the cell is recognizing, oh, I'm getting closer, I'm getting closer, I'm getting closer, because it can recognize how much of that material that's being left by the bacteria is being shed, and it's right, it's not very much far away, but it's when you're getting closer, you're warmer, you're warmer, you're warmer, right? That whole children's game, that allows for it to really find where it's supposed to be going. I wanted to play this video for you. This is a great video that I hope you can watch and listen to. Let me just turn up the volume. Okay, let's see if this will play. It won't play while I'm recording, so what I'll do is I'll add it in on the Moodle site. I'll put this video up so that you can watch it. What it is they've taken embryo from a fish, and in the embryo they are going to make a wound in the fin. And what that does is cause the recruitment of immune cells. So down on the bottom here, you have a vein. Okay. And what happens is cells actually get recruited from this vein out into the periphery due to a chemical gradient that's made at the site of infection. So the needle, right, caused the wound, and then cells out here sent a signal, and that signal is, let's say there's a protein. Maybe I need a little bigger, right? So if this protein is out here, there's a lot of it here, less, right? The further you get away, the closer you are, there's tons of it. And that gradient that's created by trapping the signals out in the extracellular matrix allows for the recruitment of these immune cells to the site of inflammation or infection. That's called lymphocyte homing. I will put that video up online for you. So that was the GAGs. That was the sugar part of the extracellular matrix. Moving on to the next part, we have the fibrous proteins of the extracellular matrix. Here are the three that I'm going to talk to you about: collagen, elastin, fibronectin. Collagen, right, demonstrated right here in the longitudinal sections, and in the cross sections you can see them. So these are perpendicular to one another. That means they're going like this in the tissue. They're creating a matrix for cells to sit down in, right? So if this were 3D, you could imagine, I don't think I can draw 3D, but the cells sit in there. So they're between and around and being held in place by these extracellular matrix proteins. So collagen, elastin, fibronectin are all proteins. Collagen is by far one of the coolest proteins we talk about. It has a particular structure. It's made up of amino acids. All right, these are on the right-hand side here. Okay, we're looking at amino acids, and they're showing you this structure GXY. So GXY is glycine, any amino acid, any amino acid. And what do you know about glycine? Glycine is small, right? It's one of the smallest amino acids. And what that does is it creates a particular structure of a single strand here. And the way collagen works is that you have 3 of these. This is called an alpha helix. The GXY makes an alpha helix, and you get 3 alpha helices bound to one another. So in panel B, in the image here on the right, you have 1 alpha helix that's red, 1 alpha helix that's green, 1 alpha helix that's blue. All right, think of these as 3 strands of string. They're alpha helices, and the 3 alpha helices wind around each other. The glycines are very small amino acids, so they don't really poke out of the structure very much, but you have a lot of the other amino acids that are prominent in the structure. So that's one really cool part of this, is the glycine, any amino acid, any amino acid. The second really cool thing is that there are modified amino acids. Modified amino acids. That means it's a normal amino acid, but it's been modified slightly. So the 2 amino acids that get modified are proline and lysine. Okay. Proline, you should remember, is the one that causes kinks. So now we're going to have a kinked protein that has a hydroxyl group added to it. Lysine is one of our positively charged amino acids, right? It's CH2 4 NH3. Is that right? Is that lysine? Yes, that's K and P for proline. All right, and they are hydroxylated, which means they have a hydroxyl group. I put this image in just to show you. This is a sequence of collagen, and I highlighted the G's. All right, there's G's everywhere. If you look, there's just G, amino acid, G, amino acid. And often it's GPK, or GKP, or PKG, or PGK. All right, so G is the glycine, P is proline, and it could be the hydroxyproline, and K is lysine, but it could be the hydroxylysine. So tons and tons and tons of this repeating structure is what allows for the formation of this particular protein. Here, why do we have these modified amino acids? Right? Why is just always a difficult one, because it happened. But if we look at the structure on hydroxylysine, here, instead of having CH2 4 times, the third CH2 group is modified with the hydroxyl. We still have the NH3. All right, so this is hydroxylysine. On the right, on the bottom here, we have the proline, right? And proline is an amino, as opposed to an amino. Amino means N like that. Immuno means N, okay? And this one is linked to a carbon. All right? So immuno is this one. Amino is this one. And the hydroxyl is on, once again, here, the 3rd carbon in that ring. Okay, why do we care? We care because these particular hydroxylysine and hydroxyproline can cross-link the individual chains that are found in collagen. Okay, so here we're looking at collagen. Right, remember we have 3, let's just say A, B, and C, the alpha helices, and you can get linkages from one collagen chain to another. So, right, this might be from A to B. So intra chain, intra means within the individual triple helix structure, or you can have what's called intermolecular. So this chain, this triplet chain, is interacting. Okay, the blue triplet is interacting with the black across the chains. So this is intermolecular. Inter is between, intra is within. Okay, let's write that down. Inter is between, intra is within. Okay, if there's intracellular sports, intramural sports, it's within a college. Whereas if you have extramural sports, you play school to school. So inter is between, okay, intra is within. So these crosslinks are what make these proteins incredibly strong. Remember I told you collagen was incredibly strong. What we have now are covalently cross-linked helices. These covalent crosslinks are what make collagen one of the most sort of sought after, right? People want to get collagen injections because collagen is a strong structural protein. They resist tensile forces. Do you remember what tensile was? Compression, right? So if I have my little square here, compression was when, right, you're pressing the two things together. Tensile was when you're pulling things apart. So the collagen really is great at keeping tensile forces from ripping things apart. It's like having a really strong rope. Interestingly, the deficiency in vitamin C that occurs sometimes in sailors, or at least it did a long time ago. All right, in sailors, they were out at sea for so long and they didn't have any fresh fruit, and so it was determined that they had this vitamin C deficiency. They don't make, all right, they don't make the modified amino acids. And that's because vitamin C is a cofactor for making those particular amino acids. The disease associated with this scurvy, if you think of scurvy, sometimes people think of pirates and their teeth are falling out. Well, teeth are held in to their sockets due to the tensile forces of extracellular matrix molecules. If you don't eat enough vitamin C, you can't make hydroxyproline and hydroxylysine. You can't make collagen very well. It's a wimpy collagen. That wimpy collagen can't hold your teeth in, so your teeth just fall out. Okay? And sometimes these sailors were called, the way people fix this, they were called limeys, because what the British naval forces decided was, okay, we're going to send these people out to sea with a ton of limes on the boat. Okay, they can, all right, I don't know if they were making margaritas or if they were just eating limes as is, but these limes helped save the sailors from getting scurvy. Okay, scurvy was not a good disease. It caused all sorts of problems with skin, with teeth, with eyes, with everything. So preventing scurvy was very important. So the limeys were what came out of the finding that vitamin C was an important cofactor for making these modified amino acids. All right, that's collagen. Elastin is very similar to collagen. And what I'm showing you in this image on the right top is, this is an aorta. Aorta. The aorta is the largest vessel, all right, that comes out of the heart, and it's this huge pump. And so you can imagine every time your heart beats, what happens is this aorta is being squeezed, right? So it's being squeezed like this around. And so this particular structure has to be very elastic. And what could you imagine might happen if that elasticity is no good? Well, let's say it pumps and it pumps and it pumps, well, and it gets weaker and weaker and weaker. What unfortunately can happen is that the aorta can rupture, and that leads to very fast, virtually 100% sudden death. So elastin is an extracellular matrix molecule that's found on the outer, right, extracellular side of structures that need to be elastic. Almost all of your organs in your body need to have some elasticity. Think about your stomach. As you eat, it gets larger. What's the purpose of the lap band these days? The lap band prohibits, all right, it actually minimizes the amount of stomach that can be utilized, right? That little bit of stomach can be expanded, but you don't want to keep expanding and expanding and expanding. It would defeat the whole purpose of having a lap band. All right, your bladder has to be elastic so that over the course of time it can fill with urine and it doesn't burst. All right, your heart is very elastic. Your organs, right, are pretty much enmeshed in this extracellular matrix, and this elastin molecule is very similar. It has covalently cross-linked modified amino acids in the structure of elastin. So on the top right of this image, they're showing you an elastic fiber. Now this is not, right, this is not the same as collagen. Collagen had that helical structure with three alpha helices bound to one another through intramolecular linkages and through intermolecular linkages. Now elastin's very different. It has very few, all right, these are all intermolecular linkages. So you have elastin molecules that are cross-linked with one another. And I like this picture because right on the top it's showing you, all right, they can be very balled up, and that's when things are relaxed, but in a stretch, okay, as urine is filling your bladder, as blood is being pumped through your aorta, then the molecules of elastin can stretch out to accommodate the increase in size associated with your bladder, the increase in size associated with your colon, the increase in size associated with your stomach or your aorta. And I put the picture on the bottom left here. There's a disease associated with defects in elasticity, in particular a protein that binds to elastin. So this elastin then gets covered, it gets bound by other proteins. Right? Up here it's still bound by proteins. And those proteins help protect the elastin. That protein is called fibrillin. And defects in fibrillin lead to a disease called Marfan syndrome. And sadly, people with Marfan syndrome often die of a sudden death due to ruptured aortas. Now, what does that have to do with this woman, Flo Hyman? Flo Hyman was Olympian on the USA volleyball team. I believe she played at UCLA. She was a very, very tall, thin African-American woman. And defects in fibrillin have been associated with the African-American population. And people who have this defect and the particular disease called Marfan syndrome are often tall and skinny with very long fingers. Their arms, the span of their arms is longer than it should be. Normally the span of your arms is the same width as you are tall. And for people with Marfan syndrome, it could be, if it's a 1-to-1, for normal people it might be 1.5 to 1. It's a really big difference. And that's all due to the fact that their elastin structures are not functioning properly, and it leads to changes in structure. And unfortunately, Flo Hyman died. She was taken out of a game, and on the sideline, she passed away in front of everybody. It happened very quickly, and very sad story. All right, that's 2 out of 3. Here's the last one, fibronectin. Fibronectin is an extracellular matrix protein. All right, and here is the structure of fibronectin. It's 2, right? So you see 2 proteins, N to C. They are covalently linked to one another through this structure. That's a disulfide, right? A disulfide bridge. So that means that there's cysteines on the ends of these particular proteins. And I think I really like this protein because it points out what happens when you have different domains in a protein. If you look at this, each fibronectin has a self-association domain, which means it can bind to itself. It has a collagen binding domain. So now we have two extracellular molecules that can bind to one another. It has a cell binding domain, which I'll talk about in a moment, and it has a heparin binding domain. Where do we find heparin? Heparin is part of the GAGS, heparin sulfate. Do you remember that? So now we have an extracellular fibrous protein that can link to fibrous proteins, it can link to itself, it can link to the sugar molecules of the extracellular matrix, and it can even bind to cells. Okay, and over on the right side of this image, it's showing you this cell binding domain. The cell binding domain has a particular structure called an RGD sequence. RGD. What is R? R stands for arginine. What is G? G is glycine. And D is aspartic acid. All right? It's a sequence of amino acids found in a particular part of this protein that allows it to link it to other cells. So this is an extracellular matrix molecule that's secreted. It's out in the periphery. It's out between cells, and it can link to a cell. And so what junction do we say could link to extracellular matrix? There were two of them. Two junctions that link to extracellular matrix are focal adhesions and hemidesmosomes. All right, and what's really interesting is that all of this starts to tie together. Now you can tie these proteins, these extracellular matrix proteins, to cells. The cells are linked through the junctions. The cells are linked internally to intracellular cytoskeletal filaments. In the case of focal adhesions, it's actin. In the case of hemidesmosomes, it's intermediate filaments. And all of this allows for the structure of cells in space, right? And remember, there is no space. The space is filled, but in the 3D, if you think about 3D, cells are sitting in a space. And how do they stay there? How come they don't move around? Well, they're sitting there because they're bound to either to other fibrous proteins of the extracellular matrix, or to sugars of the extracellular matrix, or to proteins that are bound to cells. And fibronectin really embodies that because it binds to all of them. That RGD sequence is really cool because it binds to integrins. Okay, and where we had integrins were in focal adhesions and hemidesmosomes. And I think you might remember they have an alpha-beta structure, and there were all sorts of alphas and betas, and these were the transmembrane, alpha-betas. These were the transmembrane domain proteins of focal adhesions, and there were a whole bunch of other proteins. So these alpha-betas stick out of cells and on the intracellular side. On the extracellular side, they're going to interact with fibronectin. On the intracellular side, they're going to interact, if it's a focal adhesion, with actin, and if it were a hemidesmosome, they would interact with intermediate filaments. But RGD sequences are really important for a lot of proteins. So fibronectin is another completely different protein. It has nothing to do with extracellular matrix, it has nothing to do with junctions, but it's really important for blood clotting. Okay, so I'm trying to tie together RGD sequences and why you should care. Fibronectin is a molecule that's made from fibrinogen. So blood clotting requires fibrinogen. Fibrinogen is what we call a pre-protein, and fibrinogen gets gets proteolytically cleaved into fibrin molecules, and RGD sequences can actually be targeted by snake venom. Snake venom blocks RGD sequences from causing blood clotting in the process of fibrinogen, right? So Fibronectin isn't the only protein that uses RGD. Fibrinogen uses RGD. So this has an RGD. Fibronectin has the RGD. RGD here is going to interact with integrins. The RGD here is normally going to bind to cells, I think, by their integrins. But if a snake venom is made that blocks this binding of fibrins, it blocks the process of clotting, and you continue to bleed. Well, why is that good? Why does the snake do that? Well, the snake's trying to kill you. Yay for the snake. The snake can kill you because you can bleed out. So, integrins, all right, just not to overwhelm you, but I'm sure this is a little overwhelming. If I blow this up, okay, I show you some of the key. Okay, so RGD binding, okay, RGD binding integrins. There are a lot of RGD binding integrins, anything with a red color up here. So we have red here. Here's a RGD binding protein, RGD binding protein. Okay, RGD binding protein. Tons and tons and, sorry about that, tons of RGD binding proteins. Okay, so this RGD sequence shows up in multiple places throughout biology. And, right, why do we care? Well, I care because we do have snakes here. And being bitten by a western diamondback is going to mess up the formation, well, maybe not the formation of fibrin, but it messes up the binding of fibrin molecules to integrins. And when this is blocked, you don't clot. So this would all lead to clotting. And when this is messed up by a snake, right, this is now blocking this process, you don't clot and you bleed to death. That's a sad thing. Okay. I think that's it. I think that'll do it for extracellular matrix molecules. All right, just think of it as two different kinds. You have the GAGs, the sugar protein molecules, and then you have the fibrous proteins, and be able to talk about each of the two classes. All right, have a great weekend. Talk to you later. Bye.